Hi everyone, let's talk about transcription. So we are going to be focusing on two chapters today, chapters 17 and 18. 17 is for prokaryotic and 18 is for eukaryotic transcription. For this week, I have clumped both the chapters together for this lecture. Transcription is really the process by which RNA is made from DNA. Transcription is done in the 5' to 3' direction, so which means that the template strand is going to be in the 3' to 5' direction, because all of the bases that are going to be added are going to be complementary. So for transcription, we have this coding strand and the non-coding strand. So the coding strand is going to be that DNA strand which has the sequence of the bases on the DNA are identical to the sequence of bases on the mRNA. So this is going to be our non-template strand. And the non-coding strand is going to have complementary sequence of the bases. Obviously, the uracil and thiamine will be different based on whether it's DNA or rRNA. So the enzyme that's going to make all of this happen is going to be called RNA polymerase. Polymer is a strand or like a chain. So RNA polymerase should tell you that we're making RNA. The enzyme is going to synthesize this RNA using a DNA template. So if this is our DNA double helical strand, you can see our non-template strand, which is going to be in red, and then we're going to have a template strand, which is going to be in blue. So when we make the RNA sequence, as you will see that the RNA sequence is going to be complementary to the template strand, because that is the strand off of which it will be built. So here is your RNA strand that's going to be made, and it's going to be complementary to the template strand, which is in blue. And it's obviously going to be identical to the other strand, which is right here. And this is going to be the non-template strand. Now for transcription to take place, you require something called a promoter. The promoter is going to be a region of DNA where RNA polymerase is going to bind to start off the process. In this promoter region, you're also going to have something called a start point. And the start point is really the position on the DNA which corresponds to the first base of the RNA. So, for example, if this is that U, so this is our start point. The next thing we're going to have is going to be a terminator, which is going to be a sequence of DNA that causes the RNA polymerase to finish or terminate the transcription process. And when we talk about transcription unit, so this is going to be that sequence which is between the initiation site and the termination site, and that will be used by RNA polymerase. And this can sometimes have more than one gene, possibly. When I say, or when the book says upstream, this is going to be sequences in the opposite direction. So upstream is going to be all of the sequences that are here on this end of the, or from, for example, from the expression. And then downstream is going to be anything that's below here or on this end of the strand is going to be downstream. Primary transcript. I wanted to tell you guys about primary transcript, and this is going to be that original unmodified RNA product that's going to be made and corresponding to a transcription unit. And after the primary transcript is made, it is going to be modified, and then it's going to be used as RNA. Okay, so those are a few of the terms really that I wanted you guys to know. So transcription is going to occur by the help of base pairing, and you're going to form this transcription bubble. Okay, so similar to when we have DNA replication, you're going to unwind the DNA and you're going to open it up. You have to do the same thing here, where you're going to melt the DNA to open up this transcription bubble. And at your start site, RNA polymerase is going to come and get started, and then RNA chain is going to be synthesized in the 5' to 3' direction. This is our template strand, and this is our non-template strand. This bubble is generally between 12 to 14 base pairs, and the RNA-DNA hybrid within this bubble, which is like right here, is between 8 to 9 base pairs. The RNA polymerase actually binds to the promoter site on the DNA to form what we call as a closed complex. And once you start off the process, or you initiate transcription, you're going to open the bubble, you're going to start opening up the DNA helix, and you're going to form this open complex, as we call it. And when you have the third process, which is the process, or not the third, but the second process, which is the process of elongation, the transcription bubble is going to, keep moving up along the DNA, and the RNA strand is going to be added in the 5' to 3' direction. So the new nucleotides are going to be added right here at the 3' OH. And the third part of transcription, which is going to be our termination, is going to be at the end. And when the DNA duplex is going to reform, the RNA polymerase is going to fall off, and the primary transcript is going to be released. The RNA polymerase enzyme, which is shown here in this, donut kind of shaped structure, is actually formed of a number of different components. In prokaryotes, it's a little simpler compared to the eukaryotes. It has 2 alpha, beta prime, and then this W core enzyme that's going to catalyze the transcription, and it requires that sigma factor for initiation. So when the sigma factor kind of binds, then the enzyme will go and bind to the promoter. And hollow enzyme really means the RNA polymerase form that is ready, or that can initiate transcription. It consists of all of the 5 subunits as well as the sigma factor. So the core bacterial polymerase is about 400 kilodaltons, and it has also a C-terminal domain, which we typically call the CTD, and this is the domain of the RNA polymerase that is involved in stimulating transcription by contact with regulatory proteins. RNA polymerase can also bind to random sites in DNA, and what it does is it binds, and if it doesn't it doesn't see that it's a promoter, it will move off and go and look for other sites until it really can find that promoter sequence, bind to it, and then start the whole process. The sigma factor allows or controls this binding of the DNA polymerase the RNA polymerase and the promoter with the help of conserved sequences. And these are sequences which have a particular nucleic acid typically present at this particular location. So a promoter is defined really by the presence of shorter consensus sequences. So one of the most commonly known promoters usually starts with a purine, and it is called the Tata box. It's about -10. So -10 means it's upstream of our start site. That's why we call it -10 position. And it's TATAAT. So that is the Tata box. And there is another promoter that's at the -35 location. And this is called the -35 element, which is made up of TTGACA. So, of course, a lot of T's and A's, because the T's and A's require less amount of heat to be broken apart. And this is typically the Tata AT and the TTGACA is typically what you would find, but they might differ from at one or more position at different sites. But it is pretty conserved amongst a lot of different, genus and species also. So for the sigma factor to control binding, the promoter efficiency can be affected by additional elements also. So here is our +1, which means that this is where if there's an A here, the RNA will be made with a U. And this is the -10 site, Tata. This is the -35, which is the TTGACA. But there may also be sequences that are upstream and close by to these 2 elements that can increase or enhance the process of transcription and basically the binding of the RNA polymerase with the promoter and to get the start site going. There can be mutations. For example, there are down mutations that decrease the efficiency. So if, for example, you have the Tata box and there is a mutation there, it might decrease it. Or you might have up mutations which really have the opposite effect and increase that. So mutations in the 35 sequence can affect the initial binding, whereas actually the mutations in the -10 sequence can affect binding or the melting reaction that will open up the DNA strands and make that open complex. The structure of the sigma factor. So in the RNA polymerase, the sigma 70 changes when it associates with the coenzyme, and what it really does is it helps to or promotes the binding of the DNA binding regions and the promoter. So if you look at this you have these DNA binding regions, and then this, you see this bend here too. But what happens when the sigma factor goes, then the DNA will displace this N-terminus region that was binding to this complex and allow the DNA to come and start to bind. And so this kind of opens up the structure of the DNA, because if you remember, the DNA is wound up and it's like a jumbly mess, so you want it to be a little relieved and open up. And there are specific amino acids, for example, like these ones here, arginine, threonine, that will specifically contact the bases and will specifically touch, for example, or allow this contact between the sigma 70 as well as the DNA. So once the book goes into some details about that, which I want you to read, but once we have initiation and all of this binding, the next thing that's kind of important is how we have termination. So that process in the middle of elongation, you guys already know how that takes place, similar to how DNA takes place, where at the 3' OH you have one base coming in and you lose a pyrophosphate, and you add extend the strand by one extra base every time. So that is common between DNA and RNA. But what's different is also termination. So there have been two classes of terminators that have been identified. The first is one that is called an intrinsic terminator. It requires actually the recognition of a terminator sequence in DNA that makes like a hairpin structure in the RNA product. So as the RNA polymerase is chugging along, it hits that hairpin and it will know to move away. So these are recognized solely by RNA polymerase itself, and they do not require any other factors, so they are called the intrinsic factors. There are others that require a protein that's called Rho, and these are termed as Rho-dependent terminators. So for example, if you have here, this is a hairpin that's kind of forming, right? And so when you move along with this hairpin is formed, it's going to release release the RNA strand, release the DNA, and release the RNA polymerase. Now let's look at what a couple of these things mean. So typically, the signals for the termination are going to be within the sequence that has already been transcribed by the RNA polymerase. So termination is going to rely on the template or the mRNA product that's already been made. And sometimes what happens is you can have a read-through. So this occurs actually in transcription and translation when the RNA polymerase, or in translation the case ribosome, it ignores the termination site because maybe there's a mutation in there, and it still keeps moving forward. And similarly, to regulate this, you could have, like, for example, anti-termination, where this is a control in which the termination is prevented at specific RNA terminator sites, and this allows the RNA polymerase to read the genes and keep going beyond it. Yeah. for Rho to work, okay, so it's a termination protein, and what happens is that here is the RNA polymerase, it's moving along, right, and it's transcribing the DNA. So in comes what we call the RUT site, and RUT or RUT is basically Rho utilization site, and this is a sequence of RNA that is recognized by the Rho termination factor, and it will kind of come in, and the Rho will translocate and fit itself on the RNA and form that kind of hairpin right here, if you see. And because of this formation of the hairpin, again, the RNA polymerase is going to be released, and it will be removed from the DNA. The Rho factor is released, RNA released, polymerase released, and DNA released, and you get the closed complex back again. Now let's look at chapter number 18, and we're going to look at how some of this similar processes but takes place really in eukaryotes. Now in eukaryotes, we have the DNA. It's a lot more packaged, and it's much more tightly condensed. So obviously the chromatin has to be opened up before the polymerase can bind to the promoter. There are a number of transcription factors that are required for RNA polymerase to form that initiation complex, and these factors are called transcription factor 2, for example, for RNA polymerase, and X. And as you'll see, the X can be different letters. Okay. In eukaryotes, we have the core promoter, which is the shortest sequence at which the RNA polymerase can initiate transcription. Okay, and it's generally at a lower level than you require. And for RNA polymerase 2, it is it's really the minimal sequence at which the base cell transcription apparatus can assemble, and it includes the INR, the Tata box, and the DPE. And this is typically about 40 base pairs long. Each of the promoter will contain characteristic, these short conserved sequences, and all of the polymerases will have a class of basal transcription factors that are required. An enhancer is actually going to be a sequence that's going to be further upstream or downstream, and this kind of increases basically how, or helps really, how the RNA polymerase will come and bind to the promoter. So it's kind of nudging the process along. So if this is, for example, your promoter in the start site, an enhancer might be quite removed in this. So let's say this is your, for example, promoter region right here or here, and the enhancer is actually quite far away. And that really sometimes doesn't make sense because you're looking at a DNA strand like this. But when you think of the chromatin structure and how DNA is folded and back and forth, so you can very well imagine that this loops around like this, so that this portion may be somewhere here, and so it kind of helps the promoter and the polymerase to bind. We also have something called silencers, which are really short sequences of DNA that silence the gene, so they inactivate the gene. And then you kind of all know about housekeeping genes, which are genes that are expressed in all cells because, they're providing that basic function needed for the cell to sustain and carry out its normal functions. And then we also may have coactivators, which are factors that are required for transcription, but do not bind directly to the DNA, but they're required to kind of help the DNA to interact with the local transcription factors as well as the promoters. What are the three different polymerases? So eukaryotic polymerases are 1, 2, 3. One makes ribosomal RNA in the nucleolus. 2 will make mRNA, as well as a little bit of the heterogeneous nuclear RNA that basically it comprises of transcripts of nuclear genes that are made within the nucleus. And then 3 makes small RNAs RNAs in the nucleoplasm. All of these polymerases for eukaryotes have about 12 subunits and are big complexes of about 15 kilodaltons. And some of the subunits are very common too, you know, like all the 3 polymerases will have some. And one of this is the largest subunit in the RNA polymerase 2 actually has a carboxy terminal domain that consists of multiple repeats of a heptamer. There are non-transcribed spacer regions, and these are regions between transcriptional units. So sometimes there might be genes in a cluster, and there might be space between them that we don't transcribe or it's not useful, and that's called a non-transcribed spacer. So the RNA polymerase, for example, 1 consists of a core promoter and an upstream promoter element. Okay, so that's the UPE. And the UBF1 wraps the DNA around the protein structure to bring that core and the UPE into proximity. There's also another one, for example, the SL1, I think, that includes the factor TADA binding protein, which is TBP. And this is required for all RNA polymerase initiations. So the RNA polymerase 1, for example, will bind to the UBF1 SL1 complex, and this SL1 will have the TADA binding protein that binds to the TADA box. Polymerase 3 has 2 types of promoters. The internal promoters that have shorter sequences located within the transcription site and cause initiation to occur upstream. And then it has some upstream promoters that contain sequences that are upstream of the start site. So there are some that are within that internal and then some that are upstream promoters. There are also small proteins that are actually required for the formation of the whole structure, but they're not a part of the protein themselves. And these are called assembly factors. The TF3A and C, these 2 factors bind to the consensus sequences, and then they allow TF3B kind of to bind to the start point, and then 3F2B has the TADA binding protein and it allows the polymerase to bind. So lots of small things that are coming in to get that whole the whole pre-initiation complex going and allow the transcription to begin. So RNA polymerase 2 requires a lot of transcription factors, like I said, and some of these have a short conserved sequence, for example, which is the Y2 cap Y5, which is also known as the INR at the start point, and we know already the TADA box, which is in eukaryotes. If this is the start point here around -25 base pair from the start site. And so this is a core promoter that contains the TADA box, and then this is another promoter that does not contain the TADA box. There are the DPE, which I think I mentioned earlier, which is the downstream promoter element, and this is required for RNA polymerase 2, okay? And they do not contain a TADA box, okay? And the core promoter for RNA polymerase 2 includes the INR and then the TADA and the DPE. And it might also actually contain others. Now let's talk about the TBP, correct. So TBP is going to bind to the TADA box in the minor groove of the DNA, and it kind of forms a saddle, and there's a picture in the book that shows you how it binds. So it's going to be that component that helps to position the factor that is required for each type of RNA polymerase to bind to its receptor. So like here we have this TBP coming in with the TF3B, and so once all of these factors bind, the SL1 comes and binds here for the polymerase 1. For polymerase 3 we don't have SL1, and then for Pol2 we have the TADA binding protein, and you can see, for example, so many different factors. And for Pol2 actually it's with the TF2B D complex, whereas with Pol3 it's in the TF3B complex. So just to confuse you guys a little bit more. This actually, the RNA polymerase 2, this TFT2D consists of the TBP and 14 TBP associated factors, or TAF. These are all of those blue circles. So it's like quite big, 800 kilodalton. So the basal apparatus, which is going to be there in all of these polymerases, is going to assemble at the promoter. It can have other transcription factors bind to the complex to allow this process. And when the polymerase is going to bind, so when it's 2D, it's going to be the Pol2, and it's going to then initiate transcription. There are other transcription factors that may bind and allow this to, to allow the process to start. So for example, for the for the RNA polymerase 2, you have the TF2B that's going to come, and this is showing you how the shape is kind of forming and allowing the DNA to go in and then bind, and then the enzyme is going to move forward. Okay. And again, the elongation process is going to be the same for DNA 2. So for initiation, you're going to have the TF2B, E, and H are required to melt the DNA so that you can form that open complex. And then you also require phosphorylation of the carboxy terminal domain required for the clearance of the promoter. All right, all things are ready to go, and then the polymerase will start. And then sometimes you have phosphorylation of the C-terminal domain is also required to some promoters to end the pausing and aborted initiation. So this is the whole process and how everything is kind of going to take place of initiation process, really, because that's the most confusing, requires the most amount of, let's say, players in the whole process. And you can see that here is the RNA polymerase 2, and this is that C-terminal domain. This is our promoter, and this is how, you come in and you get started. So the RNA polymerase 2 is going to come and bind, and this serine right here is going to get phosphorylated, and this will allow then other factors, for example, to come in, or, activate start the whole process of elongation. And then you actually have another phosphorylation at this second serine, and this is that fifth serine. And then you have a seventh serine phosphorylation, and we don't know why that takes place. And then once all of these phosphorylation factors occur, they takes place, you're going to start that whole process. And then when you have dephosphorylation, you're going to have a removal of the RNA polymerase. There are also histone octamers that have to be, obviously modified during this process. And that's also what that C-terminal domain is kind of doing. It's helping or coordinating that whole process of modifying that. All right. When you have DNA damage, the genes have to be repaired, and actually it's TF2H, H for Henry, that is going to come in and allow that repair of damaged genes. And then finally we have these enhancers. So the enhancer, like we talked about a little bit earlier, was basically, it's really a second type of site that's also involved in transcription, and it typically activates the promoter nearest to itself and can be just quite a distance either upstream or even downstream of the promoter. So here is an upstream promoter, or here is a downstream promoter, and this will allow their transcription to go much faster. And the upstream activating sequences, for example, the UAS, is known in yeast to behave as an enhancer, but can only work upstream of the promoter. So, you have the promoters here, and then this is an upstream promoter. So I've given you a brief overview to get you started in thinking about the whole transcription process, and email me if you have any questions.